All right, this is lecture 4 in microbiology, and we're going to start in on talking about the metabolism in bacterial organisms. Now, I remind you that metabolism refers to all of the biochemical activity that's occurring in a cell. So we talk about biochemical reactions, and often we talk about pathways going from one original substrate to a product, and then that product is used as a substrate to form another product, etc., until the pathway ends, the biochemical pathway ends with the final product. So, as I said, metabolism is the sum of all the biochemical reactions in an organism. When we look at bacteria, that's single cell, so it's the biochemical reactions in that single cell. In general, overall, reactions can be classified as catabolic or anabolic. So catabolism is energy releasing. When the biochemical reaction occurs, there's a release of energy. In anabolic reactions, and this usually happens as things break down, as more bonds are broken. And in anabolic reactions, they take up energy. The final product contains more energy than the beginning substrates. And so they consume energy. And here's a simple way of looking at it, a diagram that suggests that if you take glucose, amino acids, glycerol, fatty acids, I should point out to you that glucose is a carbohydrate, a monosaccharide, so it could be built up. It could be used to break it down and make energy, or it could be built up into more complex carbohydrates. Amino acids can be used also for energy, but they also could be built up into proteins. Glycerol and fatty acids can each individually be used to generate energy, but they can also be built up to form triglycerides. In anabolic reactions, there's a transfer. Remember, anabolic reactions will need an energy input, and the energy is usually in most metabolic reactions or biochemical reactions that are occurring, the energy has been stored up in the form of ATP, and it gets transferred to the newly more complex molecules that are being formed. And that's what's being shown here in the middle. Here, energy is getting released from ATP. So you get ADP plus free phosphate. And the energy is being transferred into more complex molecules. As I talked about earlier, glucose can be used to make more complex carbohydrates, in this case starch, a polysaccharide. Amino acids can be used to make proteins, and glycerol and fatty acids can be used to make lipids. I mentioned specifically triglycerides, but other lipids also. No conversion in any system is 100% efficient, so a bit of the energy also gets often released as heat. When these things are broken down, the reverse happens. These are catabolic reactions. When these molecules, when I say these, I'm referring to these down here. When they get broken down in catabolic reactions, there is energy released, and the energy often is stored up in the form of ATP in the cell, so it doesn't go to waste. And they get broken down to their constituent parts or further. So, microbial metabolism refers to all the biochemical reactions that are occurring in the cell, and typically you have pathways. So you have a pathway involving some sort of substrate molecule or molecules. that are undergoing a biochemical reaction going towards the formation of some sort of product. And typically there is a specific enzyme associated with that reaction which catalyzes it. And then these products now can be used as a substrate by some other enzyme to form some other products. Let's call this enzyme E2. And so you get some other kinds of products, which in turn can be used as substrates by some other enzyme to form other products until you get to the final product. Now, these are the pathway. This is a pathway. For example, glycolysis, a pathway involving conversion of glucose. In the end, glucose is completely converted into carbon dioxide and water, but along the way it goes through several stages, including formation of pyruvate and then acetyl-CoA, and then feeding into another pathway called the Krebs cycle or citric acid cycle. So you have these pathways involving this kind of substrate, enzyme, product, substrate, enzyme, product, substrate, enzyme, product, boom, until you get to the final product. So enzymes act, and when I say act, I'm talking about catalytic action on substrates to form products. Those catalytic, catalysis, ability to speed up reactions can, be anywhere from 10 to the 8 to 10 to the 10 times faster. That's an incredible increase in speed. They're involved typically at every step. There are very, very few conversions that are not that do not have an enzyme involved. They are very specific for each reaction, and they are not used. The enzymes are not used up in the reaction. They're ready to act at the end of the reaction when you get the product. They're now ready again to act on more of the same substrate to form more of the product. And this is shown in this drawing by some artist here. What you have is a substrate, and this in green here is the enzyme. And if you recall, hopefully most enzymes, vast majority, are proteins, and they have an active site where they can interact specifically with the substrate. And so the substrate binds into the active site. You get an enzyme substrate complex, and then conversion of the substrate through the catalytic activity of the enzyme into the products here. And now, as I said before, the enzyme is now same, no change, and is available to act on some more new substrate molecules. And here you see not an artist conception, but an actual classic type of biochemical model of a molecule. It's an enzyme molecule here on the left side, and they're showing the interaction of the enzyme with its substrate in the active site of the enzyme. That's in here, in this area here. Some enzymes, not all, but some enzymes require a cofactor. This would be a non-protein molecule that is acting that interacts with the enzyme. It's called a coenzyme. And then you get fully active enzyme. Apoenzyme and holoenzyme are terms that you don't come across that often. But cofactor or coenzyme is quite commonly used. So what they're showing is that there is a requirement for a cofactor for the activity of this particular enzyme. Not all enzymes require cofactors, but many do. Some examples of coenzymes, cofactors in biochemical reactions include NAD, NADP, FAD, and coenzyme A. Notice that the ink here used is black. So that's just for your information. You don't have to memorize these. I remind you again, and I know this is a reminder because you covered it in anatomy and physiology last year. I remind you that proteins in general can be denatured, that proteins have a particular amino acid sequence, which is their primary structure. They also have repetitive patterns, which are the secondary structure. And in this model here of the native or active protein, you see there's an area of alpha helix here, and there's another one here, there's another one here, there's another one there, and there's another one there. So it has 1, 2, 3, 4, 5 areas of alpha helices. The alpha helix is, of course, a secondary structure. It folds back and forth on itself, which means that's demonstrating the tertiary structure. And if there was more than one polymer or chain or subunit here, but there isn't, there's only one. But if there were more than one, then it would have quaternary structure. If it's treated or acted on in such a way that the surrounding conditions change, things like temperature or pH, then it can lose its tertiary and secondary structure and unravel. It still has its primary structure in this example here, but because it is, and that's called denaturation, it has been denatured, and it's lost its overall three-dimensional structure, so it has lost its activity. It's now inactive. Should the conditions return into the physiological range, then it would refold into this stable, the lowest energy, most stable form. Some of the factors that can denature or change the first two can act to denature and therefore can act on the enzyme activity. I mean, and it's not all or none. I mean, initially as it slightly changes its shape, the activity is reduced. At some point it becomes zero in terms of its activity. And that can happen with changes in temperature or changes in pH. So activity can be altered by the ambient temperature, the ambient pH, but also availability of substrate will change enzyme activity. If the substrate concentration is too low, then the enzyme's obviously not going to be active because it has nothing to work on. There are inhibitors that can change enzyme activity, reduce it. Some are competitive and some are non-competitive, and I'll explain that in the next few slides. And then there is feedback inhibition, and that's quite an important way of regulating pathways. All the metabolic pathways must be regulated. They can't just act on and on and on, always be turned on or sometimes often turned off. They have to be carefully regulated, and they are. And one of the ways that metabolic pathways are often regulated is by feedback inhibition on the enzyme activity, on some enzyme. All of them, some of them, only one. It depends. Let's look at temperature. You know, typically these graphs are shown as enzyme activity on one end. How do you measure enzyme activity? Well, you look at what the enzyme does, and you measure how fast it's doing it or how quickly it does it. So enzymes act as catalysts of reactions. So one way of doing that is to look for the final product. The faster the final product is being produced, then the higher the enzyme activity, right? And as I told you, temperature can act on the enzyme to change its shape. So every enzyme will have a typical optimal temperature at which it acts. Not surprisingly, although it's not always this, but not surprisingly, this one is at 37. It's an example of an enzyme in a human cell. So that has an optimal activity at 37 degrees, which is the normal body temperature. As it goes away from 37 degrees, either lower or higher, it starts to lose its activity. It starts to have a reduced ability to form product. If, however, if you raise it high enough, you'll just fry it and it'll completely get destroyed. But if you go slightly above, and then it'll lose quite a bit of activity maybe, but if you go back to the normal temperature, then it'll increase in its activity. It'll renature. It can denature, but it can also renature. pH also has an effect on enzyme activity, and again we're looking at enzyme activity, final product, how much final product, usually, not always, but typically they look at how much final product is being produced and how fast. pH, remember, when you look at pH, it's a logarithmic scale, base 10. So pH of 6 versus a pH of 5, there would be a tenfold change. Going from 6 to 5 would be a tenfold increase in hydrogen ion concentration. Similarly, from 5 to 4, even more hydrogen ions. Eventually, you get into a pH range that can break apart the protein amino acids from each other and then it's irreversibly denatured. But here you have a pH, an optimal pH between 4 and 6. Some other enzyme would have a different, these curves are not the same for every single enzyme. Another enzyme might have a different pH enzyme activity curve. For example, enzymes that are active within the stomach have the ability to have their peak activity at a pH of 2. All right, so the curve might look like this. Substrate concentration is important. Increasing availability of substrate, increasing enzyme activity, because the enzyme has to have substrate in order to act on, right? You can't play baseball if you don't have a baseball. Once you have a baseball, you can throw it back and forth. If there's more baseballs, more people can, throw them back and forth, etc. So enzyme activity versus substrate concentration rises very quickly quite often. This is often very carefully regulated for some enzymes, but often it's just this kind of what's called a parabolic curve, and eventually levels out because there's so much substrate at this point and all the way up to here that the enzyme's active site is virtually always occupied by new substrate. Okay, let's talk about inhibition. We talked about inhibitors, and we said you can have competitive inhibition or non-competitive inhibition. Competitive inhibition refers to an inhibitor that can directly occupy the active site and therefore prevent the normal substrate from getting into the active site of the enzyme. That's I call it direct competitive inhibition. And we're not going to talk about this, but often I will talk about this. An example of a competitive inhibitor often is a molecule that has a very similar but not exactly the same structure as the natural substrate. In this case, sulfanilamide can act as the inhibitor for enzyme that can act on this molecule. And the reason is because it occupies a site but cannot be broken down, so it permanently sits there. The enzyme can't act on it because it's a different molecule in the end, but it's similar enough that it can occupy the active site. Non-competitive inhibitors act differently in terms of their interaction with the enzyme. They will bind to a site that's distant from, it's on the enzyme, but it's distant from the active site. These are often called allosteric sites, and these non-competitive inhibitors are often called allosteric inhibitors. you have to understand that binding, even if it's non-covalent binding, will cause a shape change and can, as you can see here, here's the normal shape of the active site. It mirrors the shape of the normal substrate. When you have binding at the allosteric site of a non-competitive inhibitor or an allosteric inhibitor, you get a shape change in the across the molecule. The enzyme molecule changes its shape and the active site is changed, and therefore the substrate cannot interact and fit into the active site, and therefore this is acting as a non-competitive inhibitor. Okay, the allosteric molecule, non-competitive inhibitor, is acting to change the shape of the active site. Most pathways, as I told you, most pathways are very carefully regulated. They have to be, otherwise the cell would be constantly producing a final product, even if it didn't need it because it had more than enough. Cells don't work that way. Their metabolism doesn't waste energy making products that they don't need anymore. And one of the most common feedback inhibitory mechanisms to regulate the activity of an enzymatic pathway is feedback inhibition by the final end product in the pathway. Remember, pathways act by having a substrate which is converted by a specific enzyme into a product. And here we get that intermediate product, which is acted on by enzyme 1, which is acted on by enzyme 2. So this product can be acted on by a specific enzyme, which can form it into intermediate product B here, which can be acted on, and here's a third enzyme. Some enzyme pathways are, 10, even longer in terms of number of enzymes involved. But in the end, you will get an end product. And quite often, not always, but quite often, the presence of the end product can act on the first activity, will be able to bind and act on the enzyme activity, inhibit the enzyme activity of the first enzyme in the pathway. And notice here that this final end product is acting as a non-competitive inhibitor, binding at a site on the first enzyme distant from the active site, but causing a shape change so the natural substrate can't bind. This is reversible. The reason the cell was producing this end product of this pathway is because it needs it for some other kind of, function. Maybe it's producing here an amino acid that it's now going to use to synthesize some particular protein. As it makes that protein, this particular amino acid is going to be reduced in its concentration in the cell. It's getting used up, so there'll be less and less of this inhibition. More and more of that end product is disappearing, it's being used up, so there's less around to inhibit this enzyme, and the enzyme, more molecules of the enzyme will be active and the pathway will start up again and start producing end product once more. That's negative feedback inhibition. Okay, let's talk about another type of general class of reactions, biochemical reactions. These are general terms to describe reactions in relation to their use of electrons. Okay, and again, this is review. You should have done this last year, covered it in anatomy and physiology, but I'll go through it again. In biochemical reactions, you get typically change in electron content in certain types of reactions. The loss of an electron from a molecule, and you can see here that in this case, the electron is being lost from A and it's binding onto B, right? So that B has the electron. And remember that electrons are in orbit around the nucleus. The loss of an electron is called an oxidation reaction. The gain of an electron is a reduction reaction. Not surprisingly, these two types of reactions are typically found in pairs. So one molecule loses an electron, the other one gains an electron. So it's an oxidation reduction kind of pairing. And these reactions are called redox reactions. And if you want to remember, and I suggest you do want to remember, oxidation is a loss and reduction is a gain. All you have to do is remember oil rig, and you'll hopefully remember that oxidation is a loss of electrons and reduction is a gain of electrons. Let's move on. There are very important oxidation reduction or redox reactions that are occurring in cells in order to generate energy. They use these kind of reactions in the generation of ATP. Along the way, they use some other intermediate types of molecules to carry the proteins, but in the end, typically, not all, but most cells, they're trying to make ATP so that they have a chemical storage form of energy, adenosine triphosphate, ATP. Now, what you have quite often is a shifting in hydrogen atoms during redox reactions, because the hydrogen atom will have an electron, so you're moving an electron over. But in this example of an oxidation reduction reaction, you have an oxidation, which is a loss of electrons. So electrons are being transferred over. And this molecule here can act as an electron carrier. It's NAD. It's picking up electron. And now it is NADH. Let's move on. The whole point often of oxidation and reduction reactions is to enable the cell to make ATP. And ATP is made from ADP. The D is di and the P is phosphate, adenosine diphosphate. Here it's adenosine triphosphate. Remember, there's a lot of energy in phosphate to phosphate covalent bonds. So you have a lot of energy here and here, right? You have energy here. But ATP contains much more energy than ADP because it has an extra phosphate. The energy that is generated in redox reactions is taken up in the formation of the bond to the third phosphate, in the formation, in other words, of ATP from ADP plus phosphate. There are a number of ways that cells can form ATP. There are a number of general types of overall processes that the cells can use to generate ATP. They include these: substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. I'm going to go into detail on each of these in the next few slides, but just briefly for an overview, substrate level phosphorylation involves a transfer of a phosphate from one organic molecule to another. Oxidative phosphorylation involves the processes of oxidation reduction reactions. And photophosphorylation involves light energy being trapped by molecules in the cell. We're not going to talk an awful lot about photophosphorylation because it's not that important in bacterial metabolism at all. There are a few bacteria that can carry out photophosphorylation, like blue-green algae, for example. It's called blue-green algae, but it's not an algae, it's a bacteria. Okay, in substrate level phosphorylation, you have some sort of organic molecule, and this is a shorthand demonstrating that this is some sort of organic molecule. They're just showing the carbon skeleton, so they're suggesting here that it's organic by drawing it, writing it out this way. It has a phosphate bound on it, and as I told you, substrate level phosphorylation involves the transfer of a phosphate from an organic molecule to ADP to form ATP. So this phosphate is being transferred over, substrate level phosphorylation. In oxidative phosphorylation, electrons get transferred from one compound to another. That's a redox reaction. The compound that loses the electron is undergoing oxidation. Remember, oxidation is a loss. The molecule or compound that's gaining the electron is undergoing reduction. Reduction is a gain, right? So what you have is a whole series of molecules which give up. The first one gives up electrons to the next one. Then that one gives up electrons to the next one. So it's a transfer going down, 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 until you get to the final electron acceptor, which typically is oxygen. That series of carriers is called an electron transport chain. Hopefully that's not completely unfamiliar to you. You should actually have covered that last year, but we'll go into it in some detail here. The transfer of electrons releases energy, some of which gets used to generate ATP, and that process is called chemiosmosis. Now, this shows the process of chemiosmosis. I'll get into more detail later, but they are clearly, hopefully it's clear to you, showing that we're looking at a phospholipid membrane. Okay, so that's a phospholipid membrane. All of that, I'm not going to shade in the stuff in between, but all of it's a phospholipid membrane. In fact, we're looking at the inner membrane of a mitochondria. Remember, a mitochondria has an inner membrane and an outer membrane. Embedded in the mitochondrial inner membrane are a series of molecules, protein molecules, that are able to transfer from one to the next electrons. So they are able to carry out an electron transport chain. They can pick up an electron and then donate it to the next one, and then that one can donate it to the next molecule, and that one can donate it to the next one, etc. That's a series of redox reactions. In this case, this molecule here, NAD, is delivering an electron. You see there? And it's getting picked up by that molecule, which is actually the whole thing here. Sorry. It's getting picked up by this molecule here, and gets transferred to the next one, which picks it up and gets transferred to the next one, and get transferred to the next one, the next one. So there's a whole electron transport chain. The final electron acceptor is oxygen, and the energy that's left, whatever energy is left, helps it to form H2O. Meanwhile, as these molecules are handing off an electron from one to the next in this electron transport chain, hydrogen ions are being freed up and pumped across into this area here, which is the space between the inner and outer mitochondrial membrane. And so you get an increase in the concentration of hydrogen ions in that space between the two membranes. That increased hydrogen ions, that higher concentration, represents a concentration gradient. The concentration of hydrogen ions is higher in this space than in here, out here. And they are able to move through a protein that is embedded in the inner mitochondrial phospholipid bilayer membrane. They are able to move down let me change color to make sure you see it. No. Let's get something. There it goes. They are able to move down through a protein which is called ATP synthase. It's a protein which has enzymatic activity, which you should have guessed based on the name, ATP synthase. It can synthesize or catalyze the reaction between ADP and phosphate to form ATP. So as that higher concentration of hydrogen ions, as they pass through ATP synthase, ATP is generated. And in this way, through chemiosmosis, this whole process is chemiosmosis, you have production of ATP in the mitochondria of cells. This, of course, is in a eukaryotic cell because it has mitochondria, but many bacteria can also do this. They'll have this transport chain in their membrane, embedded in their membrane. They don't have mitochondria, obviously, because they don't have any membrane-bound organelles. Photophosphorylation refers to capture of energy, again through a series of electron carriers, again called an electron transport chain, to generate ATP. Our cells cannot, and most bacterial cells cannot generate ATP through photophosphorylation because they don't have the pigments, the light-trapping pigments that are able to generate energy from light energy. They can't generate chemical energy from light energy only if they have certain light-trapping pigments, the most common of which is chlorophyll, which is green. And here you see that kind of photophosphorylation. I'm not going to get into this slide in any detail. And happily or not, this is the last slide of lecture 4.